Okay, so here we're going to talk or continue our discussion about the cell membrane and talk about the different membrane proteins and begin to talk about how things get in or across membranes. So here's some objectives for this topic. Now, in general, there are three types of membrane proteins. You have integral membrane proteins, and integral membrane proteins are going to be associated with the lipid bilayer. And then you're going to have peripheral, which is going to be associated with the bilayer through either an electrostatic or hydrogen bonds within the hydrophilic domains and the polar head groups of the membrane lipids. Now, amphitropic is going to be in the cytosol and it's going to be associated with the membrane. This is a reversible association, which is usually going to be regulated through phosphorylation, for example. So here we have our peripheral protein that's associated. We have our amphoteric protein, which is floating around in the cytosol. And then we have our integral protein, which is associated with the lipid bilayers. Now, some integral membrane proteins that are going to span the membrane are shown here. So there are different types, and keep in mind that usually it's going to be an alpha helix that's going to span the membrane, and that's to keep the more nonpolar amino acids and the more non-charged amino acids interacting with that lipid bilayer. So about 20 amino acid residues are going to make that alpha helix. Now, we have type 1, which is going to be our low-density lipoprotein, or LDL receptor as an example. Also, our insulin receptor is a type 1. And the type 1 has the amino group on the extracellular side and the carboxy terminal on the cytosol, or the cytoplasm side of the cell. And then we also have a type 2, which is basically the reverse. The type 2 has the carboxy on the outside and the amino on the inside. An example of this one is the transferrin receptor. And then we have type 3, which is an important type. This is a cytochrome P450 as an example. These are detox receptors. And so our type 3 is going to have an amino on the outside and a carboxy on the inside. However, the amino group is very short and the carboxy group on the inside is very long. Type 4 is very unique. It has 7 transmembrane domain. So type 4 receptors are going to be many of our hormone receptors that were more commonly associated, that are more well known, such as our glucagon, epinephrine, norepinephrine. They're all going to use this type 4. So it's going to have several names. The type 4 is going to be called a G protein receptor, serpentine receptor, like a snake. It's sometimes called the serpentine receptor. And then we have our glycosylphosphatidylinositol, GPI-linked proteins. And these are always going to be on the outside with the N-terminus facing at the end. And so these are going to be plasminogen activator receptors, and again, they are never going to be on the inside of the cell. Now, the type 4 receptor is a serpentine receptor because of its 7 transmembrane domains. There are 2 types. There's type 4A and there's type 4B. And so type 4A is going to be a amine on the inside of the cell, carboxy out, and type 4B is going to be a carboxy in and amine out. So the example for type B is a G protein coupled receptor, and the example for type A is a sodium potassium ATPase. Now, just a little bit about those interfaces. We have tryptophan and tyrosine, which are going to be found at the interface of your lipid and aqueous environments. And so they're going to be able to interact not only with the lipid bilayer, but they're also going to be able to interact with the aqueous cytosol or extracellular matrix. In addition, there's a positive inside rule. So our positively charged amino acids, lysine, histine, and arginine, are more common on the cytoplasm face of the membranes rather than the extracellular side of the membranes. Now for an example, here's our red blood cell. So our red blood cell has, we're showing here 2 integral membrane proteins. One is band 3, and band 3 is going to be important for that chloride bicarbonate transfer. So band 3 is the bicarb chloride transfer. Remember, this is an antiport that we talked about with respect to the red blood cell. And then we have our glycophorin, and glycophorin is important for red blood cells because it generates negative charges. And so this is going to repel cells. Okay, so don't get too crowded around a red blood cell, it'll kind of push you away type of thing. And so each of these are going to be associated in the cell with different anchoring proteins. So for example, band 3 has anchorin that is then going to be attached to spectrin. And then for our glycophorin, we have our glycophorin is going to be attached to protein 4 and actin and to spectrin as well. Now here's a picture of a GPI anchored protein. And so again, this is going to be glycosyl phosphatidylate and inositol. And it's the inositol part that is really going to anchor this protein into the membrane of the lipid bilayer. And so this is going to, again, it's not an integral membrane protein because it doesn't cross the whole lipid bilayer. Okay, so when you're going to transfer into and out of the cells, you're going to have to use a transport molecule for the most part. There is simple diffusion, and simple diffusion just basically means that anything that's very small, such as carbon dioxide, oxygen, or fat, such as lipids, can cross the cell membranes. And so you're also going to have other types of transports. You're going to have transport that's going to generate an electrochemical gradient. There are going to be transports that are going to be passive, meaning that you do not need ATP. There are going to be transports in which you need active, in which you have to have ATP in order for the transport to occur. So sometimes the passive are going to include simple diffusion, or they can have facilitative diffusion. Sometimes facilitative diffusion gets confused with our active transports because in some cases there is some ATP needed, but once you open the channel, it's a flood of things coming in, and so you don't need that much ATP in order to get the gate open. And so those types of transporters are typically going to fall under facilitative diffusion. Now the ones that are truly active transport are going to be ones that let one positive in for one positive out, or 2 positive ins for 3 positives out, those type of things where you're not getting this flood of input or output, but rather it's very, very controlled. And therefore you need a lot of ATP in order to get a lot of those molecules in or out. Now another way to transport, of course, is through endocytosis that we just talked about. You can just take things up, but everything you take up initially goes into an endosomal compartment. And so if you really do need that, you have to get it out of the endosomal compartment at some way. Now we do have membrane channels. Membrane channels are going to be 3 different types. We have our alpha type, beta, and pore-forming channels. And a channel is basically a structure that translocates molecules. Some of the times those molecules can be charged or be ions, and they're going to transport them across a membrane through an aqueous channel in the protein. So this is a transport and this is through an aqueous channel. And so alpha channels, an example of alpha channel is aquaporins that are going to transport water. A beta channel are what are used in our mitochondria and also in bacteria. So those are going to be porins on the outer membrane of the mitochondria. And we also have some membrane channels that are simply referred to as toxins, in that they just punch holes in membranes, and one such toxin is a defensin that of course will target hopefully bacteria, not our own cells. So this is an example of an alpha channel. And so typically the alpha channels are going to be homooligomeric or heterooligomeric, and here is a hetero. And it's showing that it's a hetero because it has the orange and the green subunits. The molecules are going to move through the channels down a concentration gradient. So this is a concentration gradient. So there's no energy required. Some channels are going to be specifics with respect to the alpha channels, and they can have different transport mechanisms. They can be voltage regulated. You can have to have phosphorylation of your channel protein. Calcium can be involved, etc. Now your aquaporins specifically are going to transport water into cells, and there are 10 different aquaporin proteins in humans. They are going to be part of the cell membrane as homotetramers, and the monomers are going to have 6 transmembrane domains associated with them. They are going to be important in proximal tubules as well as in collecting ducts. The loss of function of a kidney aquaporin is associated with nephrogenic diabetes insipidus. So they are going to be important in our proximal tubules and our collecting ducts. So our next one is our beta barrels. So our beta barrels are going to have beta sheets, and they form cylinders. And these are going to again be in our outer membrane, our mitochondria, and they are very common in our gram-negative bacterium. So our mitochondria develop from a gram-negative bacterium. They're also found in your outer membrane of your chloroplast as well. And then finally we have our pore formers. So in this is an example of a defensin. Defensins are peptides that our cells release when there is a need for them. And they're going to be common in areas where you get infected, such as in your skin, in your mucosal tissues. They're going to have defensins. And your cells can release them. They're just peptides. And they incorporate themselves into the cell membrane of your bacteria and create a pore. And then this pore just lets a flood of stuff into the bacteria and causes the bacteria to burst.